All right, let's jump right into today's explainer and decode the insanely complex microscopic architecture of the immune system. And we're going to do this by taking a walking tour through a bustling, highly organized university campus. Now, I know it might sound kind of strange at first, but viewing the lymph node as a giant functioning university is actually an incredibly helpful cheat code. Every single building has a specific job, and every cell is basically a student or a staff member with a highly specialized role.
So, here's our campus map for today's orientation. We're going to hit up 1. The B-cell campus, 2. The elite coaching academy, 3. The T-cell department, 4. The graduation hall, and finally 5. Why this layout actually matters.
Now, before anyone can even study, they've got to get to campus, right? Both our B cells and T cells arrive via the bloodstream. You can think of the blood as basically a road network bringing in buses full of fresh students. They actually enter our lymph node university through the high endothelial venules, or HEVs. Basically, the HEV acts as the main entrance gate to the campus. This is literally where everyone scans their ID and steps onto the grounds.
All right, stop number 1, the B-cell campus.
Out here on the outer rim of the lymph node, we've got the cortex, which is home to the primary follicle. And this brilliantly illustrates our baseline state. I want you to imagine a completely normal, super quiet classroom before any exams have even been announced. The students, our naive B cells, are just sitting around. They have absolutely no homework, and they're completely unaware of the outside world because, well, they've never seen an antigen. Everyone is just relaxed.
But, these naive students aren't just floating in a void. They're actually supported by the school's staff, specifically follicular dendritic cells, or FDCs. You can honestly think of FDCs as the classroom furniture. They're the desks, the chairs, the notice boards. Interestingly, they stain positive for CD21, making them super easy to spot under a microscope. Now, they don't fight infections at all. Their only real job is just to hold on to antigens and keep the B cells organized within the room.
Of course, in immunology, the peace and quiet never actually lasts. So what exactly happens to this sleepy, quiet classroom when a virus suddenly invades the body and triggers a massive surprise exam?
Boom, an antigen shows up. Think of this antigen like a brutally strict principal kicking open the classroom door and shouting, "The exam starts right now." Immediately, some of those naive B cells recognize the antigen. They become activated, and the entire energy of the room just shifts dramatically in an instant.
Which brings us to part 2, the elite coaching academy.
So that quiet primary follicle, it instantly transforms into a secondary follicle housing a germinal center. This is an elite coaching academy with one single purpose: to train and produce the absolute best antibody-producing cells. And notice how the academy actually splits into 2 distinct floors to pull this off. Down below, we have the frantic, crowded dark zone acting as an intense study hall. And up above, we have the light zone, which functions as an interview hall.
So, let's check out that bottom floor first. Down in the dark zone, the students are now called centroblasts. They are big, highly energetic, and dividing incredibly rapidly as they try to solve thousands of microscopic exam questions. They're undergoing somatic hypermutation, which means they are constantly rewriting their exam answers, their antibodies, trying to get them absolutely perfect. Because there's so much rapid cell division, or mitosis, going on here, it's totally packed with crowded nuclei. If a pathologist throws on a Chi67 stain, which marks actively dividing cells, this entire zone lights up heavily. That makes it look visibly darker under the microscope. And yeah, that's exactly why it's called the dark zone. Now, obviously not everybody is going to pass a study session this intense. Because these B cells are so rapidly mutating their antibodies, a lot of those mutations just end up being completely useless, or they bind really poorly to the antigen. Those failing students undergo apoptosis, programmed cell death. And to keep the study hall from turning into a total mess, the school employs tingible body macrophages. You can think of them as the dedicated cleaning staff, sweeping up and basically eating the dead B cells so the campus stays spotless.
Okay, let's head upstairs to the light zone and see how this builds. After sweating it out in the dark zone, the surviving cells move up here as resting centrocytes. They aren't dividing anymore. Instead, they're facing an intense 2-step interview panel. First, those follicular dendritic cells display the antigen and ask, "Can you recognize this perfectly?" If they pass that, they face examiner number 2, the T follicular helper cell. This one asks, "Can you actually cooperate with T cells?" Only the absolute gold medal students who pass both rigorous exams survive to become high-affinity B cells. The rest? They fail, undergo apoptosis, and meet the cleaning staff.
Now, if we step just outside this high-stress coaching center, we find a distinct structural ring called the mantle zone. This is just a ring of naive B cells that never actually made it into the germinal center. If you look at them clustered tightly around that active secondary follicle, they honestly look exactly like nervous parents pacing around outside a testing center, just waiting for their kids to finish the big exam.
And right beyond those waiting parents, we hit the marginal zone. You can picture this outer perimeter as the university's exclusive alumni club. It contains memory B cells that have already successfully graduated from past infections, mingling around with a few naive B cells. While it's definitely present in lymph nodes, this alumni club is particularly obvious if you're looking at the spleen or mesenteric lymph nodes under a microscope.
All right, moving right along to our third stop, the T-cell department.
So, leaving the B-cell campus, we walk over to the paracortex. Over here, almost everybody walking around is a T-cell. Specifically, you're going to find a very reliable ratio. CD4 helper cells to CD8 cytotoxic cells sitting at roughly 2 to 3 to 1. This is beautifully easy to remember if you imagine a strict school environment where there are roughly 2 teachers, our CD4 helpers, guiding every one soldier, our CD8 cytotoxic cells.
This department also staffs some highly specialized folks called plasmacytoid dendritic cells. Think of these guys as the campus emergency coordinators. The absolute second a viral attack is detected on campus, these coordinators grab the megaphone and unleash massive amounts of type I interferons, rapidly alerting and activating the T cells to mount an immediate defense.
Next up, Section 4, the graduation hall.
After all that intense studying, testing, and coordination, everyone who finishes their training finally arrives down here at the medulla, our graduation hall. Notice how the hall is actually split into 2 distinct areas. First, we have the medullary cohorts. These look like literal rows of successful graduating students, mostly fully formed plasma cells just ready to pump out antibodies. Sitting right between these rows are the medullary sinuses. These act as wide drainage canals, and they're filled with histiocytes, or macrophages, which essentially act as security guards. They're checking everyone and everything that's trying to leave the graduation hall.
Finally, diplomas in hand, our triumphant graduates actually leave the university altogether. They flow out of the medullary sinuses and exit through the efferent lymphatic vessel. This serves as the absolute final exit gate down at the hilum of the lymph node, sending our fully trained cells out into the body to actually go fight the infection.
And that brings us to our final section, why layout matters. So, why do we care so much about this specific map? Well, when a pathologist looks at a slide, they expect to see this exact, highly organized school layout. They want to see the cortex acting as the B-cell campus, those germinal centers showing active training, the mantle zone full of resting cells, the paracortex T-cell area, and the medulla packed with plasma cells. If this beautiful structured architecture is clearly visible, it generally means the lymph node is benign. It's just doing its job, reacting perfectly to a normal infection. But you know, there's a dark side to this. If a pathologist looks into the microscope and sees that this architecture is completely effaced, meaning those structured school buildings are just obliterated and replaced by one massive, abnormal cell population, it strongly suggests a malignancy. The campus has literally been taken over by a clone, and that means lymphoma.
And honestly, this is where having that university map in your head becomes an absolute cheat code. Now that you understand exactly where these cells live and train, those terrifying medical names suddenly make perfect intuitive sense. Think about it. A lymphoma arising from the elite coaching academy, that's a follicular lymphoma. A cancer of the waiting parents outside, mantle cell lymphoma. A tumor of the alumni club, marginal zone lymphoma. So I'll leave you with this thought. The very next time you hear a complex, intimidating cancer name, ask yourself, which specific building in the lymph node university just got taken over? Keep that campus map in your head, and I promise the immune system will never look the same.